set up. So you can see your centrosomes right here in the start of the formation of these microtubules. So it's starting to get things ready for the actual cell division process. And then the next phase is pro-metaphase. This is, pro means after, pro means before. And so pro-metaphase comes before metaphase. And so in pro-metaphase, what's going to happen, remember your DNA is contained in your nucleus if you're a eukaryotic organism. And so in order to shuffle around all this DNA, you have to get rid of the nucleus. So that nucleus is going to start basically disintegrating. Or it's the nuclear envelope specifically that's falling apart, because it's the nuclear envelope that's keeping the DNA in the nucleus. So in order to move all these pieces of DNA around, you got to get rid of the thing that's corralling them in the cell, which is the nuclear envelope. And so that's going to start to disintegrate in pro-metaphase. So here you can see the nucleus is still intact. But here you can see it's still kind of all bunched in the same region, but you can't see that solid, very clear nucleus like you can in prophase. And so also we said that the mitotic spindle has already started, making itself ready in prophase. Well, in pro-metaphase, now we have these microtubules starting to attach to those, copies of DNA that we made in S phase. And so you're getting these mitotic spindles set up, and so again you can see all the condensed chromosomes, and there's all these microtubules coming out and starting to attach to all of the DNA. And then in metaphase, this is where all of those pieces of DNA, they're going to line up in the middle of the cell. So the DNA is stained dark blue right here, so that all represents all of the DNA, all the chromosomes. And so you can see that here too, although this isn't the greatest picture, but they're all aligned in the middle of the cell. It's just their tails and arms of the chromosomes are just kind of hanging out. But they're all aligned in the middle of the cell. And so at this point, this mitotic spindle has been totally formed. So the microtubules are there, they're attached to each copy of the DNA. And so then after metaphase, and by the way, one simple trick to remember what happens in metaphase, metaphase starts with an M, metaphase is when the chromosomes line up in the middle. So middle of the cell, metaphase, something like that. Okay, so after these cells, after the DNA lines up on both sides, or in the middle of the cell, now we're going to separate those copies of DNA from one another. And so that's going to occur in anaphase. And so in anaphase, that's when those microtubules, they start shrinking, and they start going back into their home, going back into their centrosome. And so as these microtubules retract, they're pulling those two copies of DNA that were held together. Now you've got them pulling on opposite forces, so you're actually separating the two copies into different cells. So anaphase is the part where you're actually separating the copies of DNA from one another. And so in anaphase, you can see actually these chromosomes being pulled apart. So this is, one copy of all the DNA, and this is another copy. And so when you look at a cell, you can physically see the copies being pulled or dragged into opposite sides of the cell. So this copy of DNA is still the exact same as this, and this copy is still the exact same as this. It's just we've separated them and put them into different cells. And so after we separate or pull apart the copies of DNA in anaphase, then we're going to enter into telophase. This is where you start to get the nuclear envelope, start to regenerate. This is where you're going to start to see this...
Furrow.
What?
Cleavage furrow.
Yeah, cleavage furrow, which is basically the place where the cell is going to kind of pinch in half. The cell's going to pinch in half and eventually make your two individual cells. Now the cleavage furrow,
That's an actual word.
cleavage furrow. Cleavage in science, in biology, I know what you think it means, but it means to cut or to pinch. So does invagination. So we have all these fun little words that we get to say. Yeah, but cleavage just means you're breaking the cell in half. Something's breaking. And in this case, we're just dividing the two cells apart. And so that's going to start in telophase. And so after telophase, You know, mitosis is over. Telophase is the last stage of mitosis. But we said the M phase is made up of mitosis followed by cytokinesis. So as telophase is kind of wrapping up, cytokinesis is going to take over, and that's when you're actually going to complete the division of the cell. And so there's two different types of cytokinesis. There's the kind that occurs in animal cells, and then there's the kind that occurs in plant cells. What we're looking at here, and by the way, these are all animal cells, and these are all plant cells. Yeah, Daniel?
What's the difference between telophase and cytokinesis?
A lot of the processes, they kind of overlap with one another. So telophase is specifically where you start to get the nucleus reformed, and the cell starts to kind of elongate. So it's the steps leading up to when you actually have this furrow, or this right before the cell starts to actually divide. So, and like I said, a lot of the times it'll start telophase, and then cytokinesis will start, and sometimes they work kind of at the same time. So cytokinesis closely follows mitosis, in particular telophase, because telophase is the last stage of mitosis. And so you can see that there's, or well, you can't see, but I'm telling you, there's two different ways that this cytokinesis can occur depending on if you're a plant cell or an animal cell. So in an animal cell, we have round cells, right? And so during cytokinesis, you're going to see this cleavage furrow start to form. And so one kind of way to kind of visualize this, imagine taking a balloon, blowing it up with air, tying it off, and then getting a piece of string and tying it in the middle. And you're going to tie it off, and you're going to tighten, and you're going to tighten, and you're going to tighten, until eventually you've got two balloons making up the larger balloon, I guess, right? That's the best analogy I can come up with. And so you're starting with one cell, but you're literally pinching the middle of it until you end up with two individual cells. That's what's happening in animal cells right here. Now in plant cells, remember they have this cell wall there. So it's not like they can just break apart, because that cell wall, it's sturdy, it's structural, it's there to support the shape of the cell. So you can't just pinch the cell in half and be done with it. So in plant cells, what's going to happen is you're going to get the formation of what's called a cell plate. So in cytokinesis in animals, you're going to have this cleavage furrow. And then in plants, you're going to have this cell plate. And this cell plate, all it is, and you can start to see, it might be hard to see, but you can kind of see this dark line starting to creep up in between the two sides of the cell. Well, this is going to be the cell plate. And so that cell plate, it'll start off and it'll basically start growing. It's working its way across until you actually end up dividing the cell in two. So one of them, we're just pinching it and squeezing it to make two cells. And in this one, we're literally dividing the one big cell by this cell plate. And that cell plate is eventually going to become the new cell wall for each of those cells. So a perfect example of a short answer question: what is the difference between cytokinesis in plant and animal cells? You would say plant cells use a cell plate and animal cells have a cleavage furrow. Simple as that. All right, and so this is kind of what we just talked about. So we have this cleavage furrow that's going to occur in these animal cells. So you can see where it's just kind of starting to pinch inward and start to pinch itself. And there's also this ring, it's called a contractile ring, that is helping, it's basically contracting too. So you have multiple forces that are coming in trying to pinch this one cell and to make it two different cells. And then in plant cells, you can get the formation of this cell plate. But again, it's kind of a process. Like you'll have all these little segments of cell plate, and then they'll eventually grow and merge out to make a new cell plate, and then that cell plate will eventually become the new cell wall of your two cells. And so when you're sending a cell into the cell cycle and you're dividing your cell, the whole point of it is to make a genetic copy. So if I have a skin cell and I want to make another skin cell, I replicate the DNA, go through mitosis and cytokinesis, And then the new cell that I made is going to be exactly identical to the first cell. It's just like taking a piece of DNA or taking a cell, putting it in a copy machine, and then getting another copy of that exact same cell. So that's what's happening when we're talking about the cell cycle and mitosis. You're making an exact genetic copy of your cell. And so it's important to regulate the cell cycle, because the cell cycle tells your cells when you need to divide. And so we don't want our cells dividing all the time. What is that? What do y'all think of when you hear of uncontrolled cell growth?
Cancer.
Cancer, right? So what do you think regulates cancer? Part of it is based on the cell cycle. So people who have cancer, a lot of the times there's something messed up with their cell cycle. So instead of a cell coming in, going around once and then leaving, it's stuck in the cell cycle. So it's going to keep dividing and keep replicating and making copies and copies and copies until you end up with this mass of cells, this tumor, that can either be harmful if it's benign, or it could be, harmful if it's malignant, and if it's not harmful, we call it benign.
How come? If
Yeah.
they know that's kind of what.
Because it's way more complicated than that.
Yeah, I know.
So that's what we're talking about, these checkpoints. So these checkpoints are there. They've evolved to kind of help make sure that this is going according to plan. And so there are various different checkpoints in the cell cycle. And so the first one that's going to happen, this is going to be the G1 checkpoint. So the G1 checkpoint, so as soon as the cell enters the cell cycle, it's going to come to this G1. Before it even can when it enters into G1, after G1 it's going to come across this G1 checkpoint. And so before the cell can move from G1 into S phase, this checkpoint is going to make sure that your DNA is good to go, nothing looks fishy, everything's going according to plan. And so once a cell gets to that checkpoint, it's literally like, being in a car. Have y'all ever seen like DWI checkpoints or something? You pull up, they check you. If you're good to go, you move on. If not, they'll stop you and you're going to have to deal with some stuff. All right, same thing here. Some stuff. And so the G1 checkpoint regulates when the cell can move from G1 into S phase. So the cell will come to this checkpoint. And by the way, these checkpoints, all they are is just a bunch of different proteins. That's all they are. Lots of proteins working together to act as a checkpoint. And so if they get the all clear signal and everything looks good, then it'll move into S phase, synthesize its DNA, and then it'll come to another checkpoint. This checkpoint is there to make sure that there's no errors when you copied your DNA, because that's what we're doing in S phase, we're copying our DNA. So it makes sense to proofread it afterwards to make sure we didn't make any mistakes. And so after it's been proofread, it'll go to the checkpoint. If it's all clear, it'll move on to the G2. If not, it'll kick out the cell, and it'll actually leave the cell cycle until it fixes itself, and then at that point it can come back in. So that's true for all of these checkpoints. If a cell ever gets to a checkpoint and something has gone wrong, and it's not going to be allowed to progress, then the cell cycle, it'll be kicked out of the cell cycle, so it'll go back to G0. Whatever was broken is going to be fixed or repaired, and then once it gets repaired, it can come back where it was and then continue to go through the rest of the cycle. And so we also have the G2 checkpoint, which regulates when you can leave G2 and enter into the M phase. And then we also have the M checkpoint that regulates when the cell can leave the cell cycle. So all of these are just kind of like proofreading mechanisms to make sure the process before was not corrupted and that everything went according to plan. And so these checkpoint proteins, there's dozens of them. There's dozens of them. So a mutation in one of them could lead to cancer, versus a mutation in a totally different one could also lead to cancer. So one of the reasons why, we said it's way more complicated than just knowing that there's a mistake in the cell cycle. But the reason why we don't have a cure for cancer is because cancer is different in every single person. The genetics behind it is so complicated and so convoluted, there's not a one-size-fits-all to cancer. Which is why, after years and years and years of research, we're kind of figuring it out, but we still got a long ways to go. And so, one example, so can you imagine, there is a gene called P53. And this P53, it's called a tumor suppressor gene. So what do you think it functions as? What do you think it does? Make sure tumors don't come up, right? And so this tumor suppressor gene, the protein that's made, P53, it's part of this checkpoint. So it's one of these checkpoint proteins. Now, almost every single, most cancers, you take a room that has 100 cancer patients, probably 90% of them are going to have a mutation in this gene. Mutations lead to faulty proteins. Faulty proteins lead to disease, right? So if you have a mutation in a tumor suppressor gene, what do you think is going to happen? You're not going to be able to suppress tumors anymore, right? And so just by one mutation in this gene, that will get this stuck so that it's making lots and lots and lots of copies of cells until you end up with a tumor. So if you kill the function of a tumor suppressor gene, you're going to get a tumor. And so this, and there's all sorts of genes like this in our genome. There's probably 20 tumor suppressor genes. This is just one example. There's other ones, same thing. There's another one called P10. Some cancer, that'll be mutated and that'll be fine. So there's a whole different arrangement of genes that can be mutated or not mutated. Plus environmental factors play a role in cancer as well. So long story short, cancer is complicated. And so it's going to take a lot of research and a lot of money, a lot of hard work to get to the point where we have a cure-all for everybody. Now, not all cancers are caused by mistakes in the cell cycle, but a lot of them are. Okay, questions on cell cycle? Yeah, okay. And so again, this is what we just talked about. I like to get ahead of myself on lectures. But, so when you have a tumor, that's going to be this uncontrolled cell growth. So you're going to have a cell that's constantly replicating its DNA and then making a copy of the cell. Constantly replicating its DNA, so it's stuck in the cell cycle. It doesn't just go through one time. It'll go and go and go and go, so you end up with millions of these cells making a tumor. And so these tumors, they can be either harmful or they can be harmless. If they're harmless, they're called benign. And so if you have a benign tumor, these guys, they're not going to be as dangerous as the malignant ones. Because the benign ones, yeah, they're still stuck in the cell cycle, but they're growing fairly slow. They're not very aggressive. They don't really have a threat of moving to other parts of your body. So these are called benign. So normally when you have a benign tumor, just go out, operation, you're done. Now, the malignant ones, these are the bad guys because these guys, they are replicating. They're like 100% in the cell cycle. So they're constantly replicating and making copies. So these guys are really aggressive. And when you have these really aggressive tumors, these malignant ones, they're capable of metastasizing. So in metastasis, what that means is the original tumor. So let's just say I have lung cancer. So I got a tumor in my lung. It's malignant, so it's very aggressive. It's constantly making new cells. Well, the bigger that tumor gets, all right, the increased chance that I'm going to have of just one of those little tumor cells either getting into my circulatory system or getting into my lymph system, which also goes head to toe everywhere throughout. So all it takes is one of those millions, billions of cancer cells to get into your blood vessels and then go somewhere else in your body, and then it'll get lodged somewhere, and then it'll start, it's still replicating, so then you get another tumor. So metastasizing is when you have a tumor in one location and it gets distributed somewhere else, and then you get another tumor. So it's the spreading of cancer is the simple way to put it. And so, again, here you can see, so this person has a benign tumor, oh, what do you know, in their lungs. And so, the bigger that tumor gets, you know, the more cells there's going to be, so the more contact it's going to come with these blood vessels and these lymph vessels, both of which go throughout all of your body. So if it gets into either one of those, it can go to your brain, it can go to your foot, it can go to your liver, it can go anywhere in the body. So you never know where these malignant tumors are going to end up. And so in this case, they ended up getting into the blood vessel or the lymph. And by the way, these cells, they are capable of squeezing their way into these blood vessels. Because these blood vessels, all they are is just cells too. And so it's capable for one cell to squeeze in between two cells that make up the blood vessel, which is kind of what's being shown right here. And so if those tumor cells get into the blood or to the lymph, again, they will metastasize, it'll make its way to some other part in the body. In this case, somewhere in the bone tissue. So what was lung cancer has now become bone cancer. Yeah.
So are these just random or?
Yeah, I mean, it's the longer you leave something untreated, the worse it can get.
Okay.
So that's why if you catch cancer in the early stages, before it's spread and the tumor's small, it's a lot easier to kill a really, really small tumor than it is one that's everywhere in your body.
So is the main tumor, is that like a host tumor?
Not really, it's just like the initial tumor.
After it's metastasized, or
after
are you
it's
able to go back and figure out which one was the original culprit, or?
You can figure it out probably just based on tumor size. The larger the tumor, the longer it's been there, right? So if it metastasizes and you only get a teeny tiny little tumor somewhere else, that tells you that it hasn't been there very long. But you definitely want to catch it before it spreads, because once it starts moving into other parts of your body, then that chemo and that radiation, it's killing dividing cells, because tumor is uncontrolled cell growth. But you're killing your normal, healthy dividing cells too, which is why you get sick when you go through chemo and radiation and all that other stuff. That's why your hair falls out, because the cells that control when your hair grows, those are dividing. So if you kill those, you kill your ability to grow hair, right? Anyways, so point is you want to catch it earlier. The earlier you catch it, the better off your prognosis will be. Other questions before we move on? Nancy, yeah?
It's kind of along with the cancer part, but what about when you have fast-growing cancers and slow-growing cancers?
Yeah.
They're both malignant. Is it just the replication?
It depends. It depends on the type of cancer. So there are some cancers, like there's a brain cancer called